Hello everyone, I'm Giulia Spagnoli, a second-year PhD student in neuroscience, and today I'm here to talk about a particular rare disease called SSADH deficiency. First of all, SSADH is an enzyme, and as you may know, an enzyme is a protein that facilitates chemical reaction. For example, it can cut a molecule, can also bind two different ones together, or modifying them. SSADH is a very important enzyme for the brain cells. It is involved in neurotransmission and in particular in the metabolism of GABA, a very important neurotransmitter. This is the reaction carried out by SSADH. We do not need to focus on the details, but you only need to know that when this enzyme is absent or does not work properly, certain toxic compounds tend to accumulate in body fluids, causing a series of neurological symptoms. Some of them are developmental delays, intellectual disability, autistic-like behaviors, and seizure. SSADH deficiency is an autosomal recessive disorder, and as you may know, we receive a copy of each gene from our parents, and for this deficiency to appear, it is necessary to inherit both mutated genes for the proteins. This disease is extremely rare, with only 450 patients known worldwide. There are no cure, but only symptomatic treatments. So the aim of my research is to fully describe a mutated protein found in a 5-year-old patient with the goal of finding a possible treatment. To achieve this, I follow 3 main studies. In the first study, I use bacteria as a factory for the production of the mutated protein in large quantities. This is the standardized method for characterizing a protein, but unfortunately, the mutation causes an alteration in the protein structure, leading to its aggregation and precipitation, making it impossible to isolate and study. The second strategy is based on the use of human cells in which I inserted the defective gene to observe how the protein behaves inside the cells. The results that I obtained from Western blot, which I'm showing you, compare an healthy protein with the mutated one found in the cells. And we can clearly see 2 bands. The one in the upper part represents the immature form, and the second one the mature form. You can notice that the mutated proteins shows only the immature form, indicating that it did not undergo all the steps to become mature. Another experiment based on the activity of the protein clearly showed that the mutated protein is completely inactive. So other experiments are still ongoing on this second part of the study to obtain more information, but now let's move on to the third strategy. The last study is the most ambitious one. I want to start from the patient's skin cells obtained through a small biopsy to turn them into stem cells. This means that they can be reprogrammed to become any type of cells in the body. Additionally, you can organize them to form an organ-like structure composed of all the parts of the organ of interest. In particular, I aim to induce them to the formation of brain organoids, which are like a small functional brain with the patient's genetic background. This is the best model to fully describe the molecular interaction of a disorder. Now I'm here in the process of completing all the tests necessary to reprogram the patient's cells into stem cells. Obviously, there is still a lot of work to do, but all these steps help me better to understand the behavior of the mutated protein in order to potentially identify a future treatment or cure for this rare disorder. Thank you for your attention.